Welcome back. In our last session, we have discussed several terminologies related to osmoregulation. So in these sessions, we are going to discuss osmoregulatory problems of freshwater and saltwater fish and some terrestrial organisms. So let's compare the physiological challenges between saltwater and freshwater fish. Saltwater fish face the biggest challenge to prevent too much water from leaving their body system. So how do they cope with this problem? Water and salt ions are usually obtained by drinking seawater. So to get rid of high salt ions but preventing water loss, saltwater fish gills contain chloride cells that are actively extrude chloride ions while passively remove sodium ions. In addition, their kidneys are modified with absence or reduced glomerulus, whereas their tubulars plays a major role in actively secreting magnesium sulfate and urea. So to conserve water, saltwater fish kidney excrete little water. On the other hand, freshwater fish face opposite problem and would like to prevent too much water from coming into their internal body system. The osmolality of freshwater is very low, around 10 milliosmol per liter. So to conserve salt water, the freshwater gills are actively absorb sodium chloride. They have relatively larger glomerulus if compared to saltwater fish. And together with the help of tubulars, sodium and chloride ions are reabsorbed in their kidneys. So freshwater fish excrete very dilute urine. Now let's look at freshwater and marine reptiles. To get rid of water, freshwater reptiles excrete very dilute urine. Marine reptiles face water shortage problem with abundance of salt ions. So to deal with this problem, sea turtles, for example, eliminate excess water through salt glands. Many marine birds also excrete extra salt in their bodies through nasal salt glands. They sneeze or shake off the salt through their nostrils. How about reptiles that live on dry land with a very little water availability? They excrete the waste in the form of semi-solid uric acid, thus reducing water loss. They have subterranean activities and usually active only during the night. Their skin are protecting them from excess water loss through evaporation. Kangaroo rats live in deserts with a very dry climate and very limited vegetation. So they eat mostly dry seed with a very high carbohydrate contents. Kangaroo rats have dry mouth and nasal passages. So they have very efficient kidneys with very long loops of Henry that are able to produce 5 times more concentrated urine than human. So these adaptations allow them to survive in desert without drinking. They also demonstrate behavioral adaptations to limit water loss. So one example is they are nocturnal and will burrow underground during the hot day and active during the night when conditions are cooler. So in summary, we have discussed osmoregulation problems faced by different types of organisms and how they cope with these challenges. So I hope you enjoy this video.